Hi, this is from Vijayakumar, Department of Physics, SNS College of Engineering. Today I am taking the topic of case study on advanced microscopes. So the microscope is a very important tool to magnification of the smaller object to the form of bigger to use of some medical and research purpose. So the topic we discussed today, the lower classes we have discussed in the optical microscope and lab microscopes. So we start from today is an introduction of microscopes. The microscope is an instrument designed to produce a magnified visual or photographic image of object too small to be seen with a naked eye. This is an instrument that can be used to observe small object, even cells. So this is an optical microscope, especially in lab purpose, and this is the next level, that means electron microscopes. So the two picture will be clearly shown in what is a optical microscope and what is an electron microscope. So from this two picture we have identified what are the difference from these two. So the optical sources we used in the light microscopes and the normal lenses we used in this microscope, but the electron microscope is in a high resolution formation because we used in a source is an electron beam, is a narrow beam. So the fall at a point to the using of condensed lenses is a electromagnetic lenses. So the divergence and convergence to the form of the electron beam we get in a very accurate magnified object we get from in a photographic plate or the screen. So this is a advantage level to compare in the light microscopes. Then the next one, this is a lab microscope. So we have check each and every microscope to the form of a lab to the advanced level. So the magnifications only upon the 1000 to 1500 x. So the less depth of the field, it involved complex lighting requirement. The resolution is in a 1 microns. But the next level of microscope electron is the greater magnification, about 1 lakh times of magnification to compare an optical. So the greater depth of field, the greater resolution is a form of 1 nanometer. So we start from the nano ranges to take a report from using of this microscopes. So this is the starting of the research microscope, the electron. The next one we discuss SIM. SIM means is a scanning electron microscopes. Here we use in only for the research purpose. So this picture we know that the only surface and in depth and the surface level modification, this all to be find from using of this SIM. The magnification range from is 20x to 30,000x. That means the compare of the electron microscope, it becomes a 20x to 30,000x. Then the resolution is forming 50 to 100 nanometers. So is in a high resolution to compare in electron microscope because it becomes scanning of the range of the surface level. The next one is in a TEM. The TEM is in a transmission electron microscopes. So in depth resolutions is high range to the form of this types of microscopes. So look at the picture of the surface, right? Each and every the surface level to be identified in a different morphology. So the magnification about in a 10 lakhs times and the resolution is 200 nanometers. The next one is in a very advanced level. This is the final stage of electron microscopes. So STEM, that means scanning, transmission, electron microscope. This to be combined to the form of scanning, electron microscope and transmission electron microscopes. So this is a very advanced level of microscope to using of the research purpose. So we have identified the topology, morphology in the resolution range, thickness, everything we have taken from these types of microscopes. The magnification about 10 lakhs times, and the range is in a 20 nanometer of resolutions. So it's in a high range of resolutions. Then this is a picture of scanning electron microscopes, and this has become transmission electron microscopes. So here to the form of primary and secondary electrons to be forming. So the primary is in a narrow beams form that we get in only for the electron microscope, but the advanced level it's forming the scattering of the electron. So the scattering of the electron we get in an exact range of the morphology. Then the stem also to be formed in the same category. This three picture is an instrument to the form of lab. So the lab to the first one is in a SEM, then second one is a TEM, and third one is in a STEM. So this three is in a picture of device in a three category of electron microscopes and future of the advanced microscopes. The image hanging on the development of the system that enable as a user of precisely and confidentially visualized sample in 3D formation, that is a feature of advanced in microscopes. Whenever we doesn't know that the result of the surface and other types of the material we have taken from this types of resolution from using of these microscopes, finally we get clear assumption of the material nature and the surface level. So the very important use of this is in a research purpose. So this is all the reference I prepared from this PPT. Thank you very much. We meet the next topic from another one of case study. Thank you.